You brought up Strand, so I think it's a good time to talk about Strand. Tell us what you guys are doing.
Strand is a next-generation genetic medicines company. We build next-generation messenger RNA therapeutics coupled with programming technologies, which allow us to actually deliver mRNA therapeutically to different targeted areas through the body. So our whole piece of how we're building these drugs are more potent, more selective, more specific genetic medicines to solve targeted diseases. One of the biggest limitations with genetic medicines and messenger RNA to date has been this inability to really deliver them to targeted areas within the body. We've had, successes with mRNA vaccines, but they're just injected into the muscles in your arm. We've had a few successes with delivery to areas like the liver, which is where a lot of things tend to accumulate when you inject them into the bloodstream. And we haven't really moved beyond that. And so we needed to find better ways to do that. That's what we're working on here at Strand.
When we were talking last time, it was really fascinating when you were talking about the abscopal response. Tell me a little bit about that, and did it surprise you?
So, to back up, right, our first drug is a messenger RNA drug that is that goes into solid tumor cells and then causes that tumor to send out a signal that stimulates the immune response. And lots of people have tried this. Even Moderna had tried this. BioNTech had tried this. And people outside of the mRNA space had. Well, what we hadn't really seen thus far was drugs that could really robustly not only activate, if you inject into one tumor, and not only activate a sort of immune response and killing and destruction of the tumor that your drug injected into, but actually is able to train the immune system to go out and fight the other tumors and take them out. And that's what's referred to as the abscopal response. So while we had a lot of data going into the clinical trial, and we have an incredible team here of scientific researchers where we believed that we had everything that we needed to see an abscopal response. It's, with drug development, you really don't know till you go into patients. I think what surprised us the most was how early in the clinical trial we saw it, right? In clinical development, you start a trial with sort of the patients who are in the worst shape, right? They've been through multiple different drugs and then even multiple clinical trials before they get to, your first in human sort of trial. You don't go into a first in human trial with a whole lot of options. And so you have very advanced disease and not a whole lot of hope. What we saw in that trial and what was really sort of shocked us last summer when the data started coming in was the extent of the responses. And so how much these patients were responding when they, had in a lot of cases not responded to anything else, right? Whether that was Keytruda, one of the best-selling drugs in the history of the world, or, some other type of clinical trial, patients who had little to no hope left, responding to this drug and responding in an aggressive way. I think that is surprising to anyone. There's no amount of preclinical mouse tumor data that can convince you that your drug is going to have that sort of a response with late-stage patients.
One of the things that we were talking about is how you have to challenge dogmatic thinking. Can you talk about that and how you're doing that at Strand?
Scientists, we view ourselves as these incredibly data-driven people who always look at things 100% objectively and let the science and the data lead us down. Unfortunately, scientists are, they're still human, right? Not unfortunate that scientists are human, but the unfortunate side of being human is that you're not going to escape that and become 100% objective just because you're a scientist. And so the past always colors the perception of the present. In drug development, for instance, they'll say, intratumoral drug, injecting things directly into tumors doesn't show abscopal responses. It shows it, in this case, but since it doesn't show it, that means it can't show it. And those are two different things, of course. It's like, our inability to do something up until now is not a prediction of whether or not that thing is possible. It's an indicator of how successful we have been at doing that thing, right? We haven't proved an impossibility. We've just proved how hard something is. I'd say that when we first started the company, we found all of this sort of dogma very frustrating, whether it was, about intratumoral drugs, whether it was about what the challenge of messenger RNA is, right? The big thought on messenger RNA was that its challenge was delivery. And our approach was that while delivery is certainly one of the things we need to work on, getting the mRNA actually into the cells that we care about, the thing that we care about at the end of the day is the expression, right? That's sort of this, to use a very, very overused term, the first principles thinking, which is, what do we care about at the end of the day? We care about the mRNA's expression, how much it expresses in the tissue that we're targeting, how much it expresses or doesn't express, hopefully, in the tissues that we want to avoid. And so that design and that build out of messenger RNAs in that sort of a way, people just weren't willing to hear it. They just kept saying, well, with delivery is the only solvable thing, or lipid nanoparticles only go to the liver, so you'll never be able to solve it. And those are fundamentally not scientifically true, but you have to fight through that dogma. You have to fight through people's preconceived notions. And sometimes you just have to, abandon hope with certain folks and just say, okay, well, I'll just have to prove that it does work this way, right? I really wish that there were better ways to sort of challenge various different dogmatic thinking within science, but unfortunately we run into it quite a bit.